All right, so we're going to talk about carbohydrates or carbs. Um, and really, carbohydrates are a type of sugar molecule. So we're going to be talking all about these guys. These guys get a bad rap these days, but we need them. They are key for our energy. So they're the main source of energy. And we're going to see this when we talk about cellular respiration. That is the main thing that a mitochondria is going to break down. Main source of energy. And they are made of carbon, hydrogen, and oxygen. And the way that they almost always are is it's one carbon for every two hydrogens and one oxygen. So carbons and oxygens will be in the same number or very close to the same number, and then there'll be twice as many hydrogens as that. So I'll give you some examples of that as we go. So these guys, I'm not going to draw these structures of these different monosaccharides that you know. Monosaccharides is the monomer. There's one of these, one sugar molecule. And then we're going to put monosaccharides together to make disaccharides or polysaccharides. And so we're going to link them together. But a monomer is the one unit, and it's called a monosaccharide. You do not have to know the structure of any of them. You do not ever have to draw them, but you should be able to look at a picture of a molecule and recognize that this is a carbohydrate, this is a lipid, this is a whatever. So as we go, you should be able to do those things. For these guys, all their structures are on page 27, or figure 2.9, so you can see them there. So glucose is a six carbon sugar. And its function is it's the major source of energy. We break down glucose in our cells. Energy, okay? And then you're going to look at fructose, which is a 5-carbon sugar. And you might guess that fructose is the sugar in fruit. Hey. So it's actually going to taste a little bit sweeter than glucose tastes. Have you already noticed that all these guys end in OSE? So sugars, generally speaking, end in OSE. That's how you can tell it's a sugar. Deoxyribose. It is a 5-carbon sugar, and this might sound familiar because it is the sugar found in DNA. It's the D in DNA. And ribose is also a 5-carbon sugar. It's this deoxyribose is obviously missing an oxygen, and ribose has that oxygen, and it's the sugar found in RNA. So you can see sugars are everywhere, and so we're going to need sugars to do jobs in our cells. So then let's look at what is sucrose. Sucrose is also known as table sugar. Like literally regular sugar, table sugar. And it is a disaccharide. So it is 2 of these linked together. In fact, it's a glucose and a fructose linked together. And we link them together by what's called a glycosidic bond. And it's a type of covalent bond, and we're going to look at that in a little bit. And then another type of disaccharide, both of these are disaccharides, a sucrose and a lactose, is the sugar found in milk. And so some people are lactose intolerant because they lack the enzyme to break down this sugar. And so it sits there and it causes all kinds of problems with the bacteria in your gut. And this one, lactose, happens to be a galactose molecule and a glucose molecule linked together. So there you are. Now, what happens if you link a bunch of sugar molecules or a bunch of monosaccharides together? You get different, so these are polysaccharides or long chains of monosaccharides. And there's some pictures of them, again, in your book. It's figure 2.10 or page 28. And so you can get cellulose, which makes up cell walls in plants. That's why plants can't move, because they have these thick cell walls made of cellulose. It's also very hard for us to digest. So cellulose is just a bunch of glucose molecules linked together. So I'm not going to draw it because, again, you can just look it up in your book. Starch is how glucose is stored in plants. So think about what's a starchy thing? A potato, right? And so that is how that plant is storing all of its sugar it stores it in its root system. So there's a lot of starchy vegetables because that's where they're storing their sugar. And cellulose and starch are just basically a bunch of glucoses linked together. They're just linked differently, so they make different molecules. And then there's glycogen, which is how we store glucose, how glucose is stored in animals. So if we have a little bit excess sugar, we store it as glycogen, so it's readily available in our muscles, and we can quickly break it down. However, if we eat too much sugar, then we begin to actually store it as fat. And that's kind of always the problem, right? So let's think about what is the role of glucose or carbs. And so carbohydrates, really, they're created by producers. Through photosynthesis. They start our food chain. That's the beginning of the food chain. They provide energy. So this is, like, you guys know this, if you drink a Gatorade, which is just pretty much pure sugar, it's going to give you a boost of energy. If you have a piece of candy, it's going to give you a boost of energy. It's maybe not the best choice in this situation because it's going to give you a high of energy, and then because your body can't regulate it. But it also gives, so another role is it's a storage of energy. So it provides quick energy, but we can also store energy this way. And we can also provide structure. So the examples are going to be our cellulose for our cell walls. Another big polysaccharide is a molecule called chitin, which is found in the exoskeletons of arthropods. So like any crustaceans like crabs and lobsters and shrimp and all those guys, that's what their exoskeletons are made out of. And then also it's found in the cell walls of fungus. So again, a structure one. So let's look at how do we make them and break them. So in order to make them, we do dehydration synthesis. It's joining of two molecules. Or compounds. So we actually do this when we're making amino acid, like linking amino acids together, when we're linking nucleic acids together, when we're linking carbohydrates together. But here's kind of the gist. I'm going to draw a really, really ridiculously simple diagram. There is a nice, lovely picture on page 27 of dehydration synthesis. They're showing you making a sucrose molecule. But pretend here this is a molecule. They never look like circles. Please ignore my really bad chemistry there. I'm going to apologize to my dad, who's an organic chemist. I'm going to hate that I'm drawing molecules this way, but we're going to do it anyways because we don't want to spend a lot of time. But you can see there's an OH group sticking off of this ring and an OH group sticking off of this ring. And so what's going to happen is you can see, look what I just circled, H, H, and O. Oh, what does that make? Water. So now and then there's this O left over. So this O is going to become in the middle. And depending on whether it's up or down, it makes these different polysaccharides that we looked at over here. And look, we also ended up with an H2O molecule. So I dehydrated this molecule. I removed water from this molecule when I joined them together. Now the opposite is when I want to break something down. So let's say I eat a potato and I now have this long polysaccharide. I should draw these these are much, much bigger than this. This is just one little oxygen molecule. This is 6 carbons and 5 oxygens and a bunch of hydrogens. So I am drawing this out of disproportion. So I want to break this apart. And so I dehydrate it. Or I'm sorry, I hydrate, like it's hydrolysis. So I'm actually going to add a water molecule in here. So we're going to do plus H2O. And let's just say we're going to break this guy off, okay? So we're going to kind of focus in on this molecule here. And we're going to break it apart. And so this one of these hydrogens is going to go right here. Oh, you can't see that, can you? Let's fix that. So we're going to break apart just this section right here. So one of these hydrogens is going to go right here, and that oxygen stayed on. And then I'm going to have my other big carbon ring, and the other oxygen and the other hydrogen are going to go right here. Okay, and so now I have two monosaccharides. So let's say this was a big glycogen molecule, and I needed to use it to make ATP. So I needed the glucose. So now I've made a glucose and a glucose. I can send those to my mitochondria, do cellular respiration, make ATP, do the work of my cell. So that's why I would do hydrolysis. So here's the question: should you eat if you're dehydrated? Question. So think about what you're doing when you're eating. So I'm going to have you answer that, and we can discuss that in class. Should you eat if you're dehydrated?